All right, this seems like a good time to get kicked off. We want to make sure we save lots of time at the end for questions. So I want to say thank you all for joining us for another MassMEDIC virtual program. I am Rachel Robinson, Vice President at MassMEDIC, and we're absolutely so thrilled to see all of you virtually again. It's been a really fun, interesting experience, kind of putting together all these virtual programs for the last 18 months, and we're excited to be partnering again with our friends at Sunrise Labs for another fantastic program. So I do want to draw your attention to a couple quick things. The Q&A option at the bottom is the best way to send questions in throughout the presentation. We'll also be saving some time for Q&A at the end, but feel free to put those in real time. And the speakers and I will keep an eye on those. And you can also use the raise your hand option if you'd like to ask your question live. If it's a little too complicated to type, you can do that as well. And just a reminder, this program is recorded, so we will be able to share this with you afterwards. So I guess without any kind of further ado, I will say welcome to Adam Jacobs, CTO at Sunrise Labs, for this outstanding program. Adam, take it away.
Thank you, Rachel. Thank you all for coming, and we're glad that we have the opportunity to be in such a great community of medical device people and be able to share the wisdom that we gain from developing many medical devices. I feel particularly warmly toward sensitivity analysis. I found it to be a tremendously helpful thing during my career at times, and just felt that it was I've had such positive experiences with it that I wanted to share that with the community. And it's particularly relevant also at Sunrise. So I do get to see, as Chief Technology Officer, I get to see many projects. We have typically about 25 at a time. And I get to oversee that we're meeting the needs of our clients, both technically, business, and medically. So it's a great position to be in, and I feel blessed to have that opportunity. So if that's helpful to you, then I'm hoping to be able to share that. So who is Sunrise? We've been around since 1991. We're about 110 people right now. We're a full service engineering development company. We don't do contract manufacturing, so we tend to focus on complex systems. Our original roots were in complex electronics and firmware. We've expanded. We have a full team now with electronics, numerous kinds of software, user interface design, firmware, machine learning, control, all kinds of stuff that you need for medical devices. We have a mechanical group, we have optics people, we have a user experience group, we have quality and a software systems group. So we get to see a lot of interesting projects, and that's a great place to be, because you start to gain some wisdom about what doesn't work. And in general, Sunrise is a great place to work. I will share that we were voted the best place to work in Sunrise in New Hampshire this year. We're just outside of Manchester, New Hampshire, about an hour north of Boston. So, we're kind of proud about that, not because we're proud of who we are, but it makes it a great place to work. So why sensitivity analysis? And why do I think this is something that's relevant? We've all probably been involved in having a project that's not doing what it's supposed to do. We're not meeting some kind of performance requirements. Examples of those might be an in vitro diagnostic test, and I've used sensitivity analysis numerous times on such diagnostic tests. So the accuracy of the test might be too low, or the repeatability might be too low. It's pretty common. So what do you do, and how do you attack the problem? But it's applicable to many other things. For example, you might have an endoscope and the image quality is not good enough. How do you make it better? I have quite an extensive background in machine learning classifier development, so have used that extensively to try to understand machine learning behavior, because machine learning is often a black box to some people, and this is a way to probe your black boxes in machine learning or in classifier development. I think of it as classifier development. So for example, if you're making a pathology slide reader, where you're scanning slides and trying to determine if there's cancer or not, or some other pathology, and you're having sensitivity or specificity issues, you can try to see what real life inputs are affecting your classifier development. Or maybe you'll have a surgical robot and your response time is variable. Why? Why is that happening? So these are the kind of problems that come up. And to the company, it's a big problem. So the leaders of the company say, get to work. And the development team comes together and they look for all the possible root causes that could cause these things. And so then they all get together, and it turns out that they say that everything matters, and they all start to point towards various things, and they're trying to figure out kind of whose fault it is and who should fix it. So that there's still a big problem because you don't really know what to do. And so as a goal of sensitivity analysis, the wisdom that we try to gain is knowing what to focus on and what to ignore. And that is a big wisdom because, as I will show you, let's say in some examples, you can spend a lot of time on things that don't matter and fix a lot of problems that don't need to be fixed, and spend less time on the really, really important ones. So I'll go back to the in vitro diagnostic device. I've done this numerous times. It's not accurate or repeatable enough. You're getting results on you're trying to read maybe the ketone level in blood or urine, and you put in you're getting variable results depending on maybe it's a test strip, and the test strips are giving variable results, and it's not accurate enough. So this is what your team looks like. It's not accurate enough, and the software guy thinks that it's the optics because there's stray light and that affects the result, and the optical engineer thinks it's the chemistry because he keeps seeing various lots giving different results, and the chemist thinks it's electronics, and the EE thinks it's mechanics, and the ME thinks it's the manufacturing, and the manufacturing thinks it's software. So that's not terribly helpful, and it doesn't particularly make for a great team dynamic. And so the system engineer or the PM looks a bit like this. He's trying to figure out what to do. So after you get your team together and make a list of things, and they say, well, these are all the, imagine you're in this meeting, and your team comes up with these possible causes of your inaccuracy. So they discuss, and they all seem reasonable, they all seem possible. But what do you do? The system engineer knows that all these things can affect the result, but the question is what to do and what not to do. You've got these 8 or 9 or 10 possible things, and it's the system engineer's job to figure out what to do. So I like to pontificate a little bit, and I'm often heard saying that a good question is worth 3 answers, because often people ask the wrong question. So, and that's true in many, many things, in many, many fields. Make sure if you're going to spend 7 years doing a dissertation that you ask the right question. The question here isn't what matters, it's how much it matters. So if something is only a small effect, then you don't want to spend time on it. If something is a big effect, you do want to spend time on it. So, and finally, sensitivity analysis will tell you, which things matter. That's the value of it. So, and then once you figure out how much things matter, you can decide what to work on. And not only that, you can use the data that you gain to determine metrics for how well the system is working, and other metrics that allow you how well it points towards certain solutions. And I'll give you an example of that later. So again, we had all these problems. You had your meeting with the whole team, and they put this whole big list, the same list as before. And now you've done your sensitivity analysis. Just imagine this for a second. Let's just imagine that everybody, is doing all this finger pointing we talked about. And you come into a meeting, and what you've done after your sensitivity analysis is you say, okay, wait a minute, everybody. I've done my work. Let's look at the results. And so you start working on, I guess I missed the the next slide. Sorry. So, after, I just want to point out that there are something called the cognitive biases. There's many cognitive biases. And one of them is that when there's a narrative that holds together, then people become overconfident that that is an actual causal result. A non-technical example of this might be that, you've all heard this in the financial markets where, the market goes down and some guy on TV says, well, there was an earthquake in Tuvalu, and so the stock market went down. And, that kind of makes sense because they make oil or whatever. And so, but I want to make the point that a good narrative does not equal causation. There's lots and lots of good narratives that are possible. And that's what all those ones that we talked about, there's an engineer and he says, well, I mean, that I know that if my power supply varies, that this is going to change and that's going to change. That's probably it. And there's a cognitive bias towards when the narrative works, you kind of latch on to that. So most people jump to conclusions too quickly. And this is a way to make sure that doesn't happen. So now you're back in your meeting and you pull up this graph. You do a sensitivity analysis and you quantify how much each of the root causes matter. Now, and let's say you've got all these root causes and you've decided that one example is the biggest one is lot-to-lot variability on the disposables. Well, you've done a test somehow or other, and sure enough, 36% of the variability is just shown from lot to lot, and then another one is just inside of a lot. There's another 21%, but there's other things. It turns out the person who brought up the idea of pipetting accuracy that affects 18-19% of the variability accuracy, or temperature, and then you can sort of see that there's quite a number of them that are in the single digits, low single digits, maybe even sub single digit, and those matter less. So, after you've done all this and, you say, well, maybe sometimes before you show this graph, you say, no, don't worry about signal processing. That doesn't matter. Don't worry about optics. That doesn't matter. Work on this one. And, it's when you can do that and back it up, people think you're either a genius or a wizard, but mostly you've done your job and done a good sensitivity analysis. So, and they haven't done the sensitivity analysis. They don't have that intuition that you have. You have an intuition about how much things matter and how much things don't. So, in a way, you kind of are a wizard. You know something that they don't do. And by the way, I like the word wizard. It seems like a good thing to do. So, what is the process for sensitivity analysis? And I'm going to take a little time on this slide because there's a lot here, and these are the things the steps that you have to do. So the first thing you have to do is determine what you're going to measure, which is the figure of merit. In the classifier, the cancer classifier, it might be sensitivity and specificity. Those are the figures of merit. It might be your RSC curve. In an IVD, it might be the accuracy of the test. In a robotic surgical device, it might be repeatability of the positioning of the surgical tool. In an optical, maybe an endoscope, it might be an optical parameter like ability to focus and how good the focus is across the field of view. So that's your figure of merit, and it's a really important thing because, again, asking the wrong question leads you to the wrong thing. So you really have to know the right thing that really, really matters to you. Like we said before, you hypothesize the root cause for the reading inaccuracy, like that graph that we looked at before with all those 10 possibilities. So the next step, now what do? The secret here is to design experiments that allows you to isolate the root cause isolate the effect of each root cause. And so you have to make an experiment that allows you to do that. And that's called a perturbation analysis. I first came across this in the optics world, because if you look at optical software, optical design software, such as ZMAX or Oslo, or there's a number of them, they've got this methodology built into their software. And what it allows you to do is, optics are great because it's really a very good model. You can really trust that the performance of a lens will be what it says. It's basic physics. We've done a good job at understanding what affects that. And it's all kinds of little things that you wouldn't think of, being a non-optics person. Like, for example, every lens has a curvature. Every lens has a kind of glass. Every lens has a variability in that kind of glass. And each one of those lenses is kind of offset a little bit or tilted a little bit. And there's all these kind of variables that go into it. And if you have a complex lens, which we did, we were building a device to scan the skin and determine if someone had melanoma. It turned out that all that had to be quite good. We needed really sort of a perfect lens, because all that variability caused loss of sensitivity in the device. So we got it. I didn't really want to become a lens optics expert, but it turned out that making this lens was important. So we did these analyses, and each one of these parameters, what you do is you take the whole system, and there's an equation for that, and you can turn each knob individually. Say, well, what happens if I take this lens and I move it up 20 microns? What happens? Or what happens if the glass has a little bit of less uniformity, or the index of refraction is a little off by this much? What happens to the figure of merit? And so that's basically the idea. It's called perturbation analysis, and you tweak each one of your variables independently of the other ones. Now, once you have a way to do that, and we'll talk more about that, because there's a real trick to that. That's not so easy. In a piece of software, that's pretty easy, right? In an equation, it's pretty easy. In real life, it's not so easy. So, but the goal, the first step is the third step is to do a perturbation analysis on each individual root cause, and we'll talk more about that. So then you perform your experiments where you change things around and see how much they matter, and then you evaluate the results. Some things might give you big effects, and things might give you small effects, and you prioritize them by the scale of those effects from each of the root causes. Now, once you've done that, you get to see that you can analyze your data in different ways, but you can propose solutions. You can say, well, now that we know that the pipetting accuracy is really important, what should we do about that? Well, gee, it turns out it's not so hard to make pipetting accuracy more accurate. It turns out we have smart engineers, and you have smart engineers, and once you give them a good problem, they'll most likely solve it. Not all problems, but many of them. And some are harder to solve than others. But again, a good question is worth 3 answers. It turns out that when we figured out that pipetting accuracy mattered, we focused on it, and we made it about 10 times better. So, and that's true with 80 to 90% of the issues. So after that, you evaluate your solutions, and you have to find a way to test it, and you verify that it worked. And we'll talk. So that's the basic process, and I'll try to to share some insights about each one of those steps. So, back to the starting points of this Pareto. If this is your goal, the variables that matter are on the x-axis, and the figure of merit is on the y-axis. And in this case, we're calling it the contribution to inaccuracy. So accuracy is typically a combination of sensitivity and specificity, and sometimes that's not what you want to use. You might want to use just sensitivity. You might want to optimize one of those two. So in this case, you can define that figure of merit. So that's the first thing you do. So perturbation analysis is interesting. It really is. So it can be analytical or experimental, first of all. We discussed the optical version of it. The analytical version of this is to have either an equation or a piece of software. And experimental one is where you're going to do tests. So we perform the perturbation analysis, and you turn each knob at a time, and you see how it affects the figure of merit. And I want to point out that how much you turn the knob matters. So what'll happen is you really don't know at the beginning sometimes how much to turn the knob. Yeah, I'm feeling like I'm a little dark, so I'm going to change this a little bit. I don't know if I look dark or not. And what I found over the time is that you have to spend a little time on it, and you'll make, you'll kind of hone in on the region that matters. For example, in some things, like if there's a tolerance of something, you say, well, I can easily keep this tolerance. Maybe it's a mechanical tolerance. I can easily keep this within 5/1000ths of an inch. It's not a problem. So what you do is you try different scales informed by how well you can control something. You might know that controlling something to a millionth of an inch is not going to be possible. So, it's not really particular, but it turns out if that's what you need, that's what you need. So you'll start with what's reasonable. You might find that you can look also, you can loosen up tolerances. So you might look very big, very medium, and very small, and then you'll hone in on the region about how much to turn the knob. And you basically work on this until you get a feel for how much the inputs change the output. And each variable you have, you can do this, and you'll get a sense of what matters. And then that's another form of wisdom, where somebody says, well, like I changed this tolerance, and I'll go back to the lens thing. There was one particular lens that was super sensitive. It had to be centered to 1.5 microns, and that's how precise an answer you can get. So, so we would go to lens manufacturing. They would say, well, we can do this to 5 microns, and think it's not good enough. So, well, that's what we can do. And I'd say, well, we have to find a way to do better, because you really know what matters. And the other ones didn't matter at all. So, not didn't matter. They were 15 or 20 or 50 microns. So normal things mattered. Normal things could be used. Normal manufacturing techniques could be used. So let's talk about analytical perturbation analysis. It usually means getting an equation or a computer model, and either one is appropriate. There's lots of numerical modeling software, or I've sometimes developed it, myself, or sometimes there's equations that you can use from the literature, and you can believe them. I mean, an example is light propagation through tissue. You can do this either way. You can use this with a forward scattering model, or you can use a Monte Carlo simulation, and both of them work. So finding the right model matters, and it has some advantages. I like to say that people tend to be either analytical or empirical in their thinking, but it's really best to be both. It's just rare to find people who can do both. So if you don't have the expertise to do the analysis, sometimes it's worth it to go find that expertise. And Sunrise has done that. We've said, we really need someone who understands. I could think of 10 examples, but I don't know if I should share them. One I think I can share is, it was dynamics of balancing a very fast rotor, and it was just, it was a specialty. And we found an expert, and that helped us model some of the math for this. So in an analytical solution, you have complete isolation of variables. You can change one knob, you can change one variable, one computer parameter at a time, and see what it does. And it's great, because it really gives you complete isolation. It's one of the reasons why it's so powerful. It also allows you to do many, many tests. You can run tens of or hundreds of thousands of tests, and that allows you high resolution for your perturbation analysis. You can also run larger ranges. Sometimes it's hard to if you're going to do something experimentally, let's say it was, it might be hard to go over the complete range that you have to cover. Maybe it's altitude. You want to see how much altitude affects it. So, I mean, you can go in a chamber, but the chamber might be limited. It might not go underwater. It might not go high enough. So larger ranges can analytically can be covered. And also, you just can get statistically significant sets of data because, well, you can run as many as you want on the computer or through analysis. You are limited to the model. I mean, models all have limitations, and so you want to make sure that you use the model correctly, that the initial conditions are set, the constants are appropriate, and people often ignore the fact that distributions matter. That different kinds of variables have different kinds of distributions. So you want to make sure, like, and I'll give you an example, like on let's say a machine tolerance on a, like a part made on a CNC machining. Well, it's not a flat distribution. It's not a Gaussian distribution. Typically what happens is, from lot to lot, you set the machine up and you get some result and it's within tolerance, and that lot will be kind of clustered around, maybe you've just got it good enough and you start making them and they're just good enough. And then next time you do it, it's just good enough on the other side of the tolerance. So, that's kind of a biased distribution. So, and don't overbelieve in normal distributions. It's a common error. And also, just important variables are often overlooked in models. So sometimes you have to make your model more complex. Empirical models can be very useful. The trick here is that the variables are mixed. So how do you control? I mean, you have a machine that's got all these mixed outputs giving you an answer, and how do you know which one it is? It's not always so easy. So sometimes you have to generate a test fixture. I'll give you an example of that. There was a reflectance of it was a dipstick test, and there was a reflectance that was measured, and you couldn't use the device we were trying to make better because it had its problems. So we had to make a special device that measured the reflectance with a calibrated reflectance meter and specially tuned with polarizers and light sources and stuff to be appropriate for our example of this in vitro diagnostic test. And we knew what the accuracy of that was, so that gave us it was a NIST controlled standard, and so you have to be carefully just do your experiments. It is labor intensive. Now you're building equipment and doing tests by hand. Usually the sample sizes are smaller because how many can you do? Hundreds, maybe tens or hundreds or maybe thousands. It usually does not cover the real life range, but you can sample the real life range. My experience is that you can be sort of, often people will make extrapolations and from statistically insignificant data sets, or sometimes you get what you get and you take as many as you can and you make a decision, which is the goal. You really want to make a decision. You don't have to get the right statistics, you have to make the decision. You do need enough to average out. Sometimes if you do have an instrument that has multiple variables and you're doing one at a time, if you do enough samples, you can then they will average out. If nothing else changes but one thing changes, the other things will average out, and you can pick that out of the noise sometimes. Some things we've seen is that the supplies can be variable. You don't always have supplies. Let's say you're working with somebody and they're developing a new instrument. They might only have 2 of them and 5 disposables, because you're early in the process. So maybe they're a research group, and it's hard to get some kind of disposable or some kind of component. They're handmade, and to do empirical work, you need a lot of them. That's easier to do in a mature device that's already in manufacture. You usually get as many as you want. And of course, empirical work, shows real life results. That's something that you might not have thought of, and you might be surprised. They say that biology is surprising, and there's a lot of truth to it. So this is an example of it. And I'm showing this because you talked about pipetting accuracy. So you take a bunch of tests. That's what the second column is. Take a bunch of tests and you come up with an average and a standard deviation of those tests. So, you can sort of see how big a change in your result you get from the pipetting accuracy or every other variable. And you do this, you inject some noise into it, you see how much, and typically what you're doing is taking, I only showed the top two because I didn't want to belabor the point, but you do 10 or 20 or 30 tests for each one of these, and then you get an average. And you've averaged out some of the noise, which there is. After you've done perturbation analysis, a common thing to do is Monte Carlo analysis. And again, not anything that probably most people here don't know about. Just to go over a little bit, it is basically understanding the outcome of combinations of your variables on the figure of merit. So for example, rather than looking at each, before in the perturbation analysis, you've isolated each one. Now you want to see how they work together. And that's pretty important because you can have counterintuitive things sometimes. For example, you can have one variable that negates another. They're not necessarily independent. You can have a combination of, say, some accuracies. One goes this way and the other goes that way. Well, they cancel each other. So the worst case analysis isn't really what happens in production. You get a random distribution of those things, and how do they add up? So that's what Monte Carlo analysis is for. And you get many combinations of these statistical distributions, and you get basically a histogram. You see that that this was accuracy of something, then you're going to get a tail of accuracies. There are some outliers, but there's a normal. I mean, there's a peak, and you can sort of see what kind of accuracy you're getting with an actual expected distribution, and that's not obvious from the perturbation analysis because because that's telling you each individual thing, not the combination of things. So it's good to do both to see how much it really matters, especially when you have dependent variables or interacting variables. I want to point out in Monte Carlo analysis, again, back to distributions, it's often underappreciated how the distributions matter. You really have to understand the process of each of the variables to understand what kind of distribution you get. These are just some examples: Gaussian, biased, or flat, or binomial, Poisson distributions. And usually you can put those into your, you can put those into your model because it doesn't know. It's just going to add the variables and select variables, and you have to give it the limits. Typically you could get the limits from your perturbation analysis, or let's say it's a manufacturing tolerance, or that you're going to, maybe if it's a temperature range, because this device is going to be in a lab, but some labs, I've heard of a chem lab in a medical lab in a hospital in Italy that's in like the dungeon of a castle. Well, the temperature there is apparently cooler than usual and moister. So even though they have some air conditioning, it's still it's a different kind of temperature distribution. So you have to know your limits, and then you have to understand how to read the results. It is not always obvious, either from perturbation analysis or from Monte Carlo, what to do with it. So I want to go a little bit into an example. So forgive me for the specificity of this example, but I want to just give you a sense of really why I like it so much. This is an example of it's a reagent, and it has a color response, and you read the color response with essentially an RGB imager. So what this is testing different lots at different concentrations of the thing you're testing. Maybe it's glucose in the blood, or it can be any kind of molecule you're looking for, really. And what this graph shows you is, you can see the red, the green, and the blue that you present. The x-axis is wavelength of light. So you can see blue is in the 400s, and green is in the 500s, and red is in the 600s. And this is the response of the reagent at different concentrations thing you're trying to test. So, for example, the top lines, the blue and purple, green and purple ones, that's in a a very low concentration. Then you get down to, you'll notice on the right hand side it'll say lot 1, lot 2, and lot 3, and there's basically, so there's 3 lots at 3 concentrations, and you'll see that the red, the yellow, and the orange lines are pretty separated. They're quite separated. So at concentration 1, you have a big separation between them. That shows a big variability just from lot to lot. And these, by the way, are averages. These are averages of maybe 20 or 30 from each lot. So, so you've got a pretty big insight here about how that's the lot is affecting your results. Also notice that it also tells you, where you look at this curve matters, because, like, let's say, for example, you're looking at the green, or the red. If you're looking at a certain wavelength of light, but let's say that moves, that's a tolerance. Let's say that moves a little bit. If you're on a slope, let's say that green thing moves over a little bit, you're on a pretty big slope. So that means that it's going to be very sensitive to the wavelength of your light source, in this case, an LED. So that kind of scares you. And you say, well, how much does it matter? How much is now, you've been looking at lot concentrations, but now you look at the data and you say, wow, I realize that the LED color might matter. So how much does it matter? So now you've taken all this data, and this is a byproduct of you having taken all this data. This shows you that as the shift of the wavelength of your LED shifts, how much does it affect the percentage of the result? And it tells you. So you can sort of say, well, I want to be within a few percent, so I need say a 4 nanometer shift in my red LED, and that's an important piece of information. So, but then you might say, well, what's the optimum wavelength? Rather than just taking the wavelength we have, and well, you can calculate that too from the data. And it turns out that at different concentrations, it matters differently. And so you might want to be, typically you want to be sort of at the peak of a curve. So for example, on this one, you might look at 660 or so, and you'll notice that red curve is pretty flat there. If you move a little bit left or right, it really doesn't move. And the same thing with the yellow ones. They really don't move very much. So, you might say, what, we've got the wrong color of LED. We were looking down at a different place, but let's put it right at the top of that curve, because that's what our reagent wants. So these are peripheral. The sensitivity analysis leads you to insights about how to make your instrument better that you wouldn't get if you hadn't done all this analysis. And that this is just one example. And I could list 50. So, which is probably why I'm sort of passionate about its value, because it really does tell you things that you never would have gotten otherwise, and it's through this process. So, I don't want to belabor the point too much, and I could talk more details, but sensitivity analysis, what's the value of it? It really does provide a deep understanding of system behaviors that you would not get without it. You don't know what matters. You don't know. You don't even know all the things that matter until you start looking at the data. And it points to those finely tuned solutions. I was able to say, you know what, your LED is the wrong wavelength. Let's move it to this wavelength, and you're going to get much more stability. A conclusion that I have is that sensitivity is really underutilized. People, even if they do, experiments or design of experiments, it's often not done enough. I mean, most engineers, I shouldn't say most engineers, most people take sort of the easy way out, and the easy way out is often, this is a lot of work, and people do it normally when they have to. They have a real problem, which is why I started with having a big problem. But in general, it's a good thing to know, because you can make your instrument better. You can loosen tolerances or tighten tolerances that matter. So I really believe it's underutilized, and it causes it causes a lot of problems by not doing it. So it does take an investment in time and resources. It's not, so you can do sometimes short studies, but often when you start to look into problems, you find that it's a richer world than you think it is, but not always. Sometimes it's simple. It is good for systems level thinking. It allows you to see the bigger picture, as well as the fine tune. And that's a talent that not that many people have, being able to see the big picture and the little details. I know what wavelength the LED wants to be, or I know how much pipetting accuracy I need, or I know that in the looking at the cancer cells, that the size matters, but the position doesn't. You get to know these things. So it does take practice. I find that having done it multiple times, really, like everything else, you get better at it, and you get to start to see insights, and you learn how to do. It's not easy to do the experiments and really isolate things. And people will jump to the easiest thing often because, well, it's easier. You don't have to. We have an instrument ready to measure that, but that's the thing that you're trying to fix. So do I really want to go out and spend $20,000 for a precision piece of instrumentation that will give me a real answer? Well, sometimes. It does provide clear direction sometimes when there isn't any. And by doing that, you reduce wasted effort. And one thing that I've seen is it reduces project and organizational paralysis and noise and fingerprinting and ambiguity and people going around in circles. I had a mentor when I was younger, and he referred to it as like the flopping fish. They're making a lot of motion, but not a lot of progress. So, and it can really, once you know what to work on, and the team gets to work on that, and then that's a lot of value. So I'm a big fan. I suggest, that when it's appropriate, it can be used. And there's a lot more I could talk about, but I don't want to bore everybody. My wife is a professor and says you shouldn't talk too long. So I think that's probably enough. If there's any questions.
That was great, Adam. Thanks. So we did have a question come into the chat. Now, not being an expert, I'm not actually sure if you answered this already or not. But someone was, asking a question around the advantage over similar methodologies. And they said something, so you can read can you see the Q&A option?
I can, yeah.
Okay, so maybe you can see if that's something you can answer.
I would call this a technique for design of experiments, really. It is a design of experiment, but people have different methodologies, and they all have sort of a similar goal to come up with the root causes. This is a particular way to gain insights by interacting with the variables, and it is a form of DOE. So it's just an experiential way that I've had that of performing it, and also it's not just the experiments. It's taking it to the next step further, where you gain insights into things that you would not have been able to understand or even knew mattered. Like that LED wavelength, there was not even it was not on the radar until we looked at the data, and then things came up. So I don't know if I have a good answer to the question, other than I think it really is if there's a Venn diagram, there's overlap.
So what percentage of projects could benefit from a sensitivity analysis? Is this something you want to do on every project, or is it most projects?
Yeah, I don't think it's every project. It tends to be when there's a quantifiable merit function, first of all, that you want to optimize. And also, it's worth it. It needs it. At Sunrise, we do about 25 projects at a time. Some of them are small, some of them are big. And, some of them, I don't know, you're going to create, a GUI for something. It doesn't need sensitivity analysis, or actually maybe the user experience people will argue with that. We have a user experience team, and they go and they test the different, outputs, and they see how it affects things. So depending on how broadly you interpret it, I'd say something on the order of a third probably could value to 20, maybe 20, a quarter to a third could value to it. Sometimes some things sometimes you don't do analysis in engineering. I like to tell the team, there's analysis that is helpful, and there's sometimes you just want to say, it doesn't matter. Let's get a really big screw and screw it together.
So at what point are you really making that decision then?
That's a good question. It's often done. Sensitive analysis is often done to solve a problem. It often comes late in the game. Usually later than, people have invested a lot of time and effort maybe into a product or into a project, and it's not doing what they want. So it's often, one of these afterthoughts, whereas it probably could have not been an afterthought if they had thought about it a little more. So, but since it's a big investment, most people don't want to do it until it's needed. Usually when we do it, it's because the thing that people thought was going to work doesn't. Yeah.
Gotcha. Yeah, I think that makes a lot of sense. I will, give you everyone a last call for questions from the audience, because I know this is it's a really deep topic and you covered a ton of information. Any kind of final parting thoughts that you want to leave people with? Is there something that, I don't know, it seems like, as you said, it usually happens because something didn't work. Is there some kind of analysis that comes before the sensitivity analysis?
Yeah, I would say keep your radar open. One thing that we see a lot is that people, sometimes we have what's called a phase 0 to a project, which is the preliminary work towards that you'd like to get done before investing in doing a commercial product. And so people come to us and say, well, this is really common, right? People come to us and say, we've done this work. We're ready to, we just want you to clean it up, and we're going to apply to the FDA and start selling. And it's usually, maybe it's a early prototype, and they've gotten some indication that it's going to work. But there's always those things that aren't working. There's another cognitive bias, which is sort of a wishful thinking cognitive bias, is that the more you work on something, that it's going to go away. And so some of these problems that, I'm going back to, we worked on some diagnostic test platforms, which are kinds of ways to, look at single molecules and have very specialized, micromems or nanoparticle kind of sensors. And it sort of worked in the lab, but it turned out that lots of other stuff could affect it. And so, early on, when you had those nagging clues that something might not be all ready to go, ready for prime time, that's a good time to do it, because you really would like to do that kind of work as early as you can, and that's why we do phase 0. So let's say, for example, in this nanoparticle project, the way that they deposit the nanoparticles matters, and they were getting lots of variability, and they thought, well, we'll fix that in manufacturing, right? But how much do you have to fix it? How is it possible to fix? So during those phase 0s, it's a great technique so that you can essentially raise your technology readiness level to the point where you really want to start investing. And Sunrise has we've kind of got our own medical technology readiness level, because the military has one and NASA has one, and medical things have their own secret sauce. We don't really have to worry about being in outer space so much. So yeah, those early projects, I think, keep your eye out for those things that don't go away.
Yeah, it's a great one. So we did actually have one more question come in. Would you say that when you have a signal with a lot of noise, it could make sense to run a sensitivity analysis?
Absolutely.
Okay.
Absolutely. So again, let's talk about classifiers. You're doing a machine learning project, and you've got some data, and you worry that maybe maybe I shouldn't talk about these particular examples. I think I better not talk about those specific examples. But you're getting a signal, and that signal, maybe it's an electrical signal, and you're going to build a classifier to tell you maybe when you're having an AFib effect, and or maybe ventral. I actually worked on something for ventral fibrillation. There was an algorithm, and that was going to automatically detect when you were in VFib and sound an alarm. And great idea for certain people. And you can run your signals that you've taken through your algorithm and get the results. Well, you can just inject some noise, right? You can just say, okay, here's my signals, and luckily they're digital nowadays. So I'm gonna just randomly add X amount of noise, or I'm gonna add certain kinds of noise. There's respiration noise that will change your signal levels. People, there's impedance measurements. There's all kinds of noises that are going to happen in real life, and we often model many different kinds of noises, inject them individually into our model, and see what the result is. So I might have 5. There's one we're working on now, and it's trying to measure something in the body, and the breathing matters. So we have a breathing noise. There's just one of ref noise, which is a biological noise that probably you biomedical engineers are familiar with. One of ref noise is something that happens in the body. So we inject one of ref noise. There's just what happens if our amplifier gives us noise. That's Gaussian noise. What happens if there's thermal noise from resistors? And you just start injecting all these noises, and it's great. You start to see how much matters and how much you have to have to block that stuff out. So absolutely, Yannick, it's a great technique for that.
Great. Then that was a really wonderful question to kind of close up on. And then, so I guess I'm just going to close down by saying thank you, Adam. That was such a wonderful presentation. And as I said, it was a ton of information. And I know we're all going to take a lot away from that. So thank you so much for putting that together.
Okay. And thank you, everybody, for coming. And again, we're Sunrise Labs, and we do a lot of fun things. And maybe I'm sure that in this community, we either know some of you or hope to meet some of you.
All right, everyone, have a wonderful rest of your day, and we hope to see you again soon.